Dear friends, welcome to this edition of Basic Anatomy. Today we will discuss Chapter 3, Lecture 6, where the topic will be bone as a connective tissue. I will concentrate more on the tissue feature, that is histological features. The other general anatomy details I will cover in another chapter. Now you see, this is the generic diagram of a bone. It is cut in such a way and magnified that it presents both the transverse section as well as longitudinal section. Both the sections are there, therefore it gives a very composite view of the bone. You can see the transverse section is at the upper part and the longitudinal section has been cut out like a cake in the anterior aspect. Now the key essential aspect of this design is there are vertically running blood vessels and nerves. The red vertical lines are the blood vessels. Now around the blood vessels is the bone pattern clearly defined. Now these blood vessels and nerves run in a canal called the Haversian canal. The Haversian canal, together with its lamellae around it, is called the Haversian system. So basically we are going to look at these in detail. You can see that projecting from the transverse section is one complete Haversian system. In the middle of the Haversian system is the Haversian canal with the blood vessels. We will see the more details in the next few slides. Now this is a diagram. We are going to use this as the reference diagram and see it in actual histology preparations. Some of these may be unstained sections, but sometimes we can also decalcify because bone contains a lot of calcium. It can be decalcified and then stained with hematoxylin and eosin. Now we can recall from the previous diagram the blood vessels located in the vertically running Haversian canals. The Haversian system is the Haversian canal and its contents, along with the concentric lamellae associated with it. With this background, let us go to the microscope. Here is the scanner view of the bone, a compact bone. You can see at the surface is the periosteum and at the inner aspect is the endosteum. Basically, these are connective tissue, dense irregular connective tissue. Next, immediately underneath the periosteum or the circumferential lamellae. There are three types of lamellae: circumferential, interstitial and concentric. Concentric lamellae we have already seen. They are surrounding the Haversian canal and the whole unit forms an Haversian system. The circumferential lamellae is usually underneath the periosteum. There is a third type known as interstitial. I will cover it in the next few slides. Now see, that is a Haversian system. It contains an Haversian canal along with various bone-rich lamellae. Now one lamella is separated from the adjacent lamella by a line of a circle of spaces. The spaces are known as lacunae. I have identified this in the by a oval shaped area, that is the Haversian canal. a Haversian system. The Haversian system is also known as an osteon. You see, the core of the Haversian system is the Haversian canals. The canal contains blood vessels and nerves. Next, cutting across the bone, that is running perpendicular to the long axis of the bone and connecting one Haversian canal with another adjacent canal, or what are known as interconnecting channels known as the Volkmann's canal. They connect adjacent Haversian canals, therefore their content is also similarly blood vessels and nerves. Now this is the other third lamina which I was mentioning about, that is the interstitial lamella. while each Haversian system is a cylindrical, a vertical cylinder. In between the cylinders, there is some residual lamellar bone. That lamellar bone is called the interstitial lamellae. It acts more like a packing layer. Now you see, one Haversian system I have magnified and I have drawn lines to show you the design of the lamellae. They are known as concentric lamellae. That means all the circles have the same center, and that center is in the middle of the Haversian canal. Next, bone is deposited in concentric circles, and all of them are centered around the Haversian canal. Now there is a cement line which briefly demarcates one Haversian canal, and the immediate triangular piece of lamella between three adjacent Haversian systems is the interstitial lamella. Now you see, separating the lamella or the lacunae are the cavities in which the osteocytes are located, but these lacunae communicate with each other with adjacent laculae and neighboring lacunae by small tiny canals known as canaliculi. Now those are the lacunae, those are dark spots, and the sun ray appearance that is seen around these is the canaliculi. You see, now that's the lacuna. It appears empty because this is a stained specimen. As I told you, we will have to send it through the tissue processing during which time the cells are likely to be destroyed. So these empty spaces actually contain living cells called osteocytes. Now there are various types of cells in the bone. We are now discussing the compact bone. Basically we have three types: osteoblast, osteocytes, and osteoclasts. Now we will try to understand each of these a little more in detail. Now you see, osteocytes are the key bone cells. They are derived from the osteoblasts. As the osteoblasts deposit calcium on the ground substance, these cells get embedded within their own secretions, and the cavity in which they are embedded is the lacuna, and the cells are now known as the osteocytes. Osteocytes communicate with adjacent osteocytes in other lacunae through cell processes by which nutrition is exchanged from one cell to the other, so that the entire bone gets its supply of nutrition. Now these processes are located within the canaliculi that we discussed a little while back. Now osteoblasts are the precursor cells. It is these osteoblasts that lay down bone on a template called osteoid. They secrete alkaline phosphatase, and with that help they help in mineralization of the ground substance, and their cytoplasm is typically basophilic in nature. Osteogenic cells are basically stem cells, bone forming stem cells. They are the ones which differentiate into osteoblasts, and the osteoblasts then become the osteocytes. The osteogenic cells replenish the stock of osteoblasts from time to time. The last variety is the osteoclasts. They are multinucleated. They are larger cells. Sometimes even up to 50 nuclei may be seen in a single cell. They are bone resorbing cells. They remove bone. That is the difference between osteoclasts and other cell types in the bone. Next, you can see here the radial canaliculi connecting adjacent lacunae. This is a catechu stained specimen in very high magnification under light microscopy. Now the processes of the osteocytes, as I told you, are going into this canaliculi. The matrix is filled with hydroxyapatite crystals. The contribution to these crystals are mainly calcium phosphate and calcium carbonate. Now we will see with a few examples how this design can be better appreciated. You can see I have taken a wooden cross section from an old wood specimen. On that specimen, you can see I have arranged peanuts, the common peanut. Now, the peanut, when you examine carefully, is communicating with other peanuts. I have used the example of the wire. Let's understand it a little more in detail. The red sheath, the plastic sheet of the wire, is the canaliculus or a canaliculi in plural, and the copper wire that is in the core is equivalent to the cell processes. Now you see, we are looking at a decalcified bone. Appreciate the following in a decalcified bone. The contrast improves because there is hematoxylin as well as eosin. Therefore, the cell outlines are the lacunar outline are better seen in this section. The prominent lacunae, a large Haversian canal, and the remnants of the highly organized concentric lamellae. These are the things that we will appreciate in this particular photograph. Now you see, in the Haversian canal, the arteriole, venule and nerves. Basically, it is a canal for distribution of blood vessels. Now that was an overview of the the histology of bone. Thank you.